Yes, so we're going to talk about some methods that we're developing, looking at how we can track exchange of information between brain regions using time series data. And so the motivation for this comes back to the idea that the absolute explosion of interest in multivariate pattern analysis, and we're now pretty good at statically saying at one time point or in one brain region what sorts of stimulus distinctions can we decode and in a multivariate sense. But what we're less good at doing is knowing what that information is actually used for, whether it's exchanged between brain regions, and really for a complete understanding of something mechanistic, we want to be able to say there's this sort of information and it's exchanged in a particular way between brain regions and transformed and so on in order that we could track something from input to output, right? Also amongst the motivation is this fantastic paper from DeWitt et al. Theoretically, with the provocative title, Is Neuroimaging Measuring Information in the Brain? Where they make a very valid point that just because we can decode something as the experimenter outside of an fMRI scanner or an MEG scanner doesn't actually mean that that is information that's being used by other brain regions, right? So it's not necessarily information to the brain just because it's information to us. And they put forward this criteria that actually to be information, that information in one brain region would have to be used by another, right? So in order to be able to test this, we need to have some metrics or some ways of figuring out whether information is actually being exchanged between brain regions. So that's our question. And so we're talking about MEG data, which of course you can estimate some sources for, and you might find yourself in a situation like this. Does this point? I'll use the mouse.
Oh, it's on. Yes, it does. This points in left and right.
Aha. Thanks, Ola. Okay, so you might have some brain region, say here at the back of the brain, and when you present the participant with a particular image, you can measure the patterns of activation that you estimated for that source, and you can do that for another brain region. And then what you can do is compare that for different stimuli, right? So this yellow alien here to a red one, and you might see that the pattern changes, and that's something we can track through time. So at each time point, we can measure how different are these two patterns, right? So this is a typical decoding analysis resolved through time. But what we might like to know is, to what extent do activity patterns in one brain region actually influence activity patterns in another, right? And in both, we'd like to answer this in both directions. Now, the problem, of course, is that these patterns don't correspond to each other in meaningful ways, at least not simply, right? So we heard about this from Olaf earlier, that there might, in fact, be linear transformations between these two in terms of which voxel here maps onto which voxel there, but we don't necessarily know what they are. So this is not a straightforward kind of correlation that we can do. And so there's lots of different approaches to this, but they rely on a similar kind of idea. And the idea is what you've got to do is extract something in both brain regions that are directly comparable. The trick we're going to be using here is to summarize our patterns of activation in a representational dissimilarity matrix. So here we summarize whether the pattern for this red object is similar to this pattern for the orange object, and we give that a value, and then we summarize how similar it is to the green object to give that a value, and so on for all the possible combinations. Now this we can do for both brain regions, and now we're in a space where the two things are directly comparable to each other. So we can start to ask questions like, is this pattern here correlated with this pattern here? Which we couldn't do on the raw data, at least not in a straightforward way. Okay, so this is what we're calling information flow analysis, and we can refine the question now. So the question that we're asking is, does the structure of representational space in one brain region predict that of another brain region at a later time point? So today I'm going to talk about three possible approaches we can use to do this. The first one just uses partial correlations. So it works like this. For a particular time point that you care about, you can extract the representational dissimilarity matrix from a particular region, so say the back of the brain here. You can do that for another brain region, like the front of the brain here. And then you can also look backwards in time and say what happened in the history of each of these two brain regions. So now for a particular brain region, a particular time point, say the front of the brain at time 0, we can ask how well can we predict what the representational space there should be based on what happened previously at another brain region, right? And we can say, can we have that prediction, is that prediction any better than the prediction we would have had based solely on the history of the region itself? Right, so do we get more information if we include information about what was happening in the back of the brain at time minus 100 than if we just try and base a prediction on what was happening already in the frontal lobe? So this is using the logic of Granger causality, right? Whereas if you are adding anything to your prediction, then you can say that the occipital lobe Granger caused the pattern that happened in the frontal lobe. And this would be what we would consider sort of feed-forward information based on what we know of brain organization. We're just taking a simple partial correlation, and you can plot that then for that time point, right? Then, of course, you can do this in the other direction, and now you would be seeing what the influence of the frontal lobe is on the occipital lobe, so that would be a measure of feedback information. And you can plot that too. Then you iterate to the next time point, and so on and so forth. And in this way, you build up through time a plot of the partial correlations which reflect the feed-forward information and the feed-backwards information. Okay, so I'll show you now a couple of examples. Here's the original study where we proposed this idea. It was developed by Erin Goddard, who's now at McGill University, when she was working as a postdoc in my lab in Sydney. And the first study here, we have these 24 real-world objects. And so our RDM is just a 24 by 24 matrix, which says something about how similar the patterns of responses are to each of the different objects. And we separated things into the perifrontal and perioccipital MEG sensors. And when you do this, you see some sensible looking things. So here in blue, you have feed-forward flows of information, and in red, you have feedback flows of information, and you can directly contrast them to one another, which is what we do for inference. So you can see here that these time points labeled blue here, there's significantly more feed-forward information than there is feedback, whereas at later time points, you see more feedback than feed-forward. So sort of sensible pattern. You also see that feedback tends to happen after stimulus presentation has ended, which in this experiment was not until 500 milliseconds. And you can compare this to the decoding traces. So here you see that feedback doesn't really start until this red line here represents the perifrontal areas, until decoding is at least above chance in the front of the brain. Then you can also go just a little bit further and separate things out, because of course what information is in the RDM is the only thing that this system can pick up on. So you can mess around a little bit to get some more specificity on what's being passed between brain regions according to what you put into that RDM that you give to the analysis. So in another experiment, we have these coloured objects here, and what you can do is only build an RDM based on the colour discriminations and collapse across form. And now you'll have something that tells you about the flow of information of colour, right? So colour information flow. And when you do this, again, we have the same axes here, so numbers above 0 mean there's a dominance of feed-forward information, and numbers below 0 mean feedback. The colours work the same way. And now we have a shorter stimulus, but you see a similar thing again. More feed-forward information at the beginning of the trial, feedback happens later. You can also plot this in another way here, where now the depth of the colour here represents the height of the spike here. So the dark blue means good evidence for feed-forward information, flows of information, and the dark red means feedback information. And I just replotted it that way so that I could overlay it with the decoding. So here the black line is the occipital lobe and the green line is the frontal lobe. And again, you see that the feedback information only onsets after you at least have some information in the frontal lobe. So these sort of sanity checks check out. The other thing that's really nice about this is that you can compare the time course of your feed forward and feed flows of information to the conditions that you cared about in your experiment in the first place. So here's a decoding again, but now it's separated into attention conditions, where the red line is information about the attended feature of a target, and all the other lines are information that wasn't relevant to the task. So there was a distractor object, and there were also that distractor object and the target object had some irrelevant features. And you can see here that in terms of just pure decoding, which is shown on the y-axis, only the information about the attended object is sustained through time. And then you can compare the time course with which this feedback is happening to the time course with which you see an attentional effect, and it turns out that they're very similar. You can also do a similar thing for the shape information with the same stimuli, very similar story again. You have a different time course now, but again, feed forward first and then feed back. So I'm going backwards. And again, you see this nice concurrence between the time at which feedback tends to dominate in the system and the time at which you start to see these attentional effects emerging. And just to show you that we're not the only group doing this work, this is from Hamid Karimi in Nature Scientific Reports this year, and he does the same analysis, but now his stimulus presentation's even shorter, just 50 milliseconds. Again, you see feed forward first, followed by feedback, but his feedback then is even earlier. And the nice thing here as well is that he also broke it down so he put in his RDMs, rather than using all his objects, he separated them out by the different categories and so on. So again, trying to push at getting a bit more specificity of what's actually moving between the brain regions. So you can resolve things in this way into subcategories. So with this, you can infer these Granger causal influences of information coding on one brain region from one brain region on another, so something like information exchange, and you can characterize when the feed-forward and feedback suites dominate the flow of information. So if that's your question, then you can stop here. Of course, this is limited at the moment because it's a partial correlation. It's only working for 2 regions. And we're also averaging. So, well, I didn't explain in detail, but in that past time region, what we were doing was taking a large window and just averaging across it. So it's still fairly crude in some ways. And can we go further? Well, here's a second approach, which is based on very similar ideas, not developed by me. This is all Tim's work, Tim Keatsman here at the CBU. And the idea is very similar, but now we're going to be using a multiple regression instead of a partial correlation. So the main difference here is that when we're predicting these time zero RDMs, rather than just averaging across the time in the past, what we'd like to do is use all the time points. So actually use the RDM movie to predict the future of our target brain region. So you can do this using just a hierarchical GLM, but not combining these weights with combining using nonlinear least squares. And you can have a model that looks like this. So what you're going to be doing is trying to predict the target at time 0, this RDM, based on the source region at time minus 120 and minus 118 and minus 116 and so on, up to minus 20 in their case, plus all the same time points but from the target. Okay, so now rather than one correlation, you're doing a big GLM and you're saying how much variance can I explain if I use all these time points from my target ROI and all these targets from my source. Then what you can do is reduce it. So you say, what about if I didn't include the source, I just had my target ROI, RDMs, would that be different? Okay, so the inference here is if there is more variance explained by the full model than by the reduced model, then again you have this Granger causality logic where you're saying, okay, so then there was something predictive or there was information from the source to moving from the source to the target. So you calculate this unique variance, which is the extra variance that is explained by the full model relative to the reduced model. And then what you've got to do is compare that to something. And their proposal is to compare this to the unique variance that you would get if you did the whole thing again, but you just used the pre-stimulus, so a baseline, right? And this is important because full models have more predictors, so they're always going to explain more variance than reduced models. But that shouldn't be the case in the baseline if it's all just noise. So I'll just go over the steps for that again. First of all, you combine all your predictors for a full model. Then you combine all but your source predictors to make a reduced model. And then you calculate the difference. So your unique variance is the variance explained by the full minus the variance explained by the reduced. And then you test the unique variance across subjects against your baseline. Okay, and when you do this, here's some data from Tim and his group. So here on the y-axis you have the additional variance explained, right? So numbers above 0 means that there was either feed forward or feedback flows of information. In his case, the purple is a feed forward because it's going from V1 to 3 to up to V4 LO, and in this plot from V4 LO to IT. And the orange line is coming back the other way. And then you see here the significance tests against 0. So the major difference here is that where in my case we were only comparing feed forward to feedback, here because you're comparing against a baseline, you can have both feed forward and feedback happening at the same time, which is actually something we might expect could be happening in the brain, so it's nice to be able to see. So you have here the option to be able to see bidirectional information flow at the same time points. You also have the advantage or disadvantage, depending how you look at it, that you're not averaging over time points. This potentially would be easier to scale up if you wanted to use multiple regions rather than just 2. One more option I just wanted to quickly mention. This is published by Alex Clark this year in JICN. Here are some brain regions. And now, rather than doing Granger causality between RDMs in the way that we've been doing for both of the approaches so far, we're going to do Granger causality on model fits instead. So that looks like this. You have your brain data that might look something like this. And then you would also have a model RDM, right, which in their case was predicted from some deep neural networks, but it could be a theoretical model or whatever it might be. And then you ask how well do these correlate with each other. So this is so far a standard RSA approach. And you plot that through time. These aren't Alex's real data, but it's just to give you an intuition. So here you would have time, and here you have the correlation, or whatever metric you like to use, between the model RDM and your brain data RDM. And then you can do that for another brain region. And now here's the trick. You're going to do the Granger causality at this level, right? So now you're asking, are there Granger causal influences between this model fit and this model fit? And also, of course, the other direction. So the difference here is that now, rather than looking at whether the structure in one brain region is predicted by the structure in another brain region, you're asking whether the extent to which a structure fits a model in one brain region is predicted by the extent to which the structure fitted the model in another brain region. So the really cool thing about that now is it gives us some specificity, right? So where before you had an entire RDM and another entire RDM and you looked to see whether they could be related to each other, what you didn't have was any information about what parts of that RDM were being passed back and forth, right? Apart from to the extent that obviously only the conditions that are in the RDM could be part of it. You don't know whether, for example, animacy might be driving the difference between them, or some low-level visual features, or what. Whereas here you get some specificity because you're actually applying things to a model. So you can look, for example, to say, does the extent to which my RDM looks like my visual model predict the extent to which it looks like my visual model in another brain region, and so on and so forth. And they used visual models and semantic models as it happens. Again, it's bidirectional because you can do this in both directions at once. And what I think is most exciting about this and really cool is that you can explicitly now incorporate transformation of information. Right? So you can have something like this, where you can say, to what extent does my model fit with visual information predict my model fit with semantic information in another brain region, right? Which is something we might expect, that brain regions might not just tell each other to represent the same things, but the information might actually be transformed between them. And that's explicitly incorporated in this approach. The downside is that this is no longer time resolved. So you don't have these plots through time of when these different feed forward and feedback flows are happening. Okay, so now let's just take a minute to take a step back and think a bit more broadly about what we're doing. And I wanted to highlight some conceptual issues that we are very much in progress of working through at the moment, and don't really have answers to, but I'm glad to discuss it if anybody has ideas. So first of all, I've been trying to highlight as I go, this is kind of really important next question, right? If we could work out two brain regions are communicating with each other, it'd be nice to know what information is being fed back between them. Now, on the one hand, these RDMs are kind of hypothesis light, right? They're not hypothesis free in the sense that, of course, they're dependent on what your entries are in your RDM. They're somewhat exploratory, but of course, depending what conditions you put into them, there's only limits to types of answers that you could get out. So they're light on the hypothesis. Of course, to work, though, this thing doesn't have to be a true RDM. So in the Goddard et al. And PrEP work, we just put color discriminations in a vector and called that our RDM and did the same analyses as before. And because we only fed the thing color discriminations, then what's being passed about can only be, at least at some level, related to the color of the objects, right? So you can have this sort of intermediate thing where it's not truly hypothesis-driven and it's not truly hypothesis-free either. And then you have the Alex Clark approach where you're doing the Granger on the model fit time courses, and that gives you much more specificity. But now you're relying on your a priori hypothesis about what should be exchanged, right? So there's a kind of balancing act here depending on what type of analysis you want to do and how good your priors are. So do we prefer hypothesis light or hypothesis driven? I'm not sure. And to what extent do we really expect brain regions to drive each other towards a common representational space? Right, so this is something Tim and I have talked about quite a lot because both of our analyses are based on this idea that what the brain regions are trying to do is force each other to have the same patterns, right? But then you have the question about, well, would that really be how the brain would work? Do we actually, is the goal for all the different brain regions that have to do with your task to end up with a similar representational structure, or does that need to be the end goal actually? Or could it be something more like this, where actually you need different types of information represented in different brain regions? So I think that's open. Another question here is, what offset do we expect? Because both of these approaches rely on taking time point 0 and then looking back in time to see the causal influences, and the question is, how far back should we be looking? And we tried to base this on responses to visual stimuli in macaque monkeys and sort of scale it up a bit because humans are bigger than monkeys and guess. But if you look at it a bit more empirically, this is data from Aaron Goddard with the colour and shape analysis. You can see that actually for a wide range of windows, you seem to see a very similar answer, which is interesting. So here, each of the different colours represents a different length of delay, and the black line is the average. And you can see for this range from 55 milliseconds up to 125 milliseconds, it doesn't make a huge amount of difference. The only place where it seems to make a difference is here, where you see that the shorter time windows show effects first, which makes sense. And I think the reason why this isn't making a huge amount of difference, well, because part of me thought, well, we might find the time point that tells us it's 40 milliseconds between this region and that brain region, and that would then be an additional source of information for us. I think it doesn't work like that, partly because the whole thing's quite smoothed, right? So here are some autocorrelations through time, and you see the diagonal here is obviously, trivially, it's a correlation between a brain region and itself at the same time point. So that's ones. But then around it, you see some spreading, right? So you see that there are sort of blocks of time where the brain is relatively simple in terms of the types of information it's encoding, or in terms of the RDM structure. So that's probably why this doesn't matter too much. But if you push it right to the extreme, it does start being important. So for example, if you had 0 offset, you see this absolute dive in the partial correlation. But that's because it's a partial correlation, so you're perfectly predicting things from the past of now, basically. But there's this quite sharp function where everything from about 25 milliseconds onwards is essentially flat. So you can kill it if you push it right out to 500 milliseconds, then things start to get really wacky. Nearly finished, but a couple of other quick conceptual issues. How many regions and time points would we want to include? Again, it depends how good a hypothesis you've got of what's going on here, but we're wondering how exhaustive your search could be, like whether you could really blow this up and do it in a kind of searchlight type of way. And a couple of other open questions. How does this sort of connectivity relate to other connectivity measures? We don't know yet. And how much of this whole thing is going to turn out to be attributable to leakage, which is something that Olaf is going to help us figure out with some modelling. Because we have this idea that, so this is the setup that I've shown you. Now, what happens if there's information that's leaking from the frontal ROI to the occipital ROI here? That would tend to drive this to be a stronger correlation, give you partial correlations, where in fact it wouldn't be due to any signal in the occipital ROI, but only due to leakage. And you have the same problem in this sort of setup here. So there's a summary of the 3 different methods, which I won't go through, but I think I've said pretty much everything that's on there, but if you want to talk about it, we can. And I'll just end by saying thank you, especially to Aaron Goddard, who developed all of the IFA stuff, and to Tim, who has been absolutely fantastic for discussing all this stuff in the last few weeks, and obviously has been developing in parallel very similar ideas. Alex Clark and Courtney, who is now carrying out some modelling work for us to try and figure out what we should be seeing when we know the ground truth of what's going on in models. And Olaf, who, as I said, has been very instrumental and helpful in talking about signal leakage and how that might play out in the context of these kinds of analyses. Thank you.